So since open science is just science done right, and our institutions are not providing us with infrastructure to do science right, then we have to fight for it, don't we? I'm Björn Brembs. I'm a neurobiologist and I study the neurobiology of behavior in Regensburg. The short answer of why I work with flies and snails is because they're the ideal experimental animals to answer the questions that I'd like to study experimentally. Historically, I've never really wanted to work in neuroscience. I always thought I'm not smart enough. It's very interesting, but it's very, very complicated, and there would be no way I could ever, find anything meaningful out in this gigantic mass of neurons and tangles of axons and stuff. And that feeling started before I even started studying biology. And then I don't really recall how that happened. I ended up in a lecture by Martin Heisenberg on the neurogenetics of Drosophila. And I was very quickly hooked. He managed to start with big questions such as free will and the freedom of behavior that we have and how we make decisions, and managed to divvy the question up into small subcomponents that he convinced a lot of us that actually Drosophila can be used to study small aspects of what I thought was way too complicated. And so I started working with him, and I ended up doing my PhD with him. And now I'm not so sure if that was so smart, because even flies are very complicated. And now that I've worked with them for 25 years, maybe I should go to something even simpler. Anyway, after I did my PhD on flies, I found in the literature articles on the Aplysia marine snail, and this seemed to me like the perfect and ideal next step. I had worked on operant and classical conditioning in fruit flies for my PhD, and I thought now was the time to move beyond behavior and into the wetware, into the neurons, how they do it. And there had been a recent series of articles out of the lab of Jack Byrne in Houston, Texas. They had been working on a case of operant conditioning that I thought was the logical extension of my work that I had done in Drosophila before. And so I moved there. And snails, these snails in particular, are just fantastic. These neurons are so big, we joke that you can use a fork to record from these neurons. And so the neural level on these animals is easily accessible, and the behavior can be made very rigorous and isolating in isolating the relevant components. So that was very attractive. And flies complement snails in that they add the genetic toolbox that the snails lack, which makes studying the wetware a little bit more difficult in some cases, but offers a lot of advantages on the behavioral level. So flies and snails are awesome, and they're ideally suited to answer questions that are just too complicated at this point to study on a neuronal level in animals that are more complicated, such as mammals or birds or things like that. I've wanted to be a scientist ever since in high school I started reading books from scientists about their discoveries. One of those I'd be very interested in would be the father of my thesis advisor. And that would be Werner Heisenberg, the physicist who discovered the uncertainty principle. And I would like to talk with him about how he interpreted his findings and what he thinks about modern interpretations of quantum mechanics. Because very often today people say that a certain interpretation of quantum mechanics was developed or was formulated around that time of Heisenberg and colleagues. And I would like to ask him if that's actually correct, or if they're misrepresenting some of the developments that were happening at the time, and what he thinks about current quantum mechanic interpretations. Second scientist I'd like to meet would, of course, be Fred Skinner, because he's been working on and thinking about a lot of the questions that we have been trying to tackle with completely different animals and setups. And in fact, our approach of using neurogenetics and neurobiology is contrary to his radical behaviorist approach, when you only look at behavior and you never look into the black box that is the animal. So I'd like to ask him if our approach of actually looking into the black box to find out how the animals are doing what they're doing, if he would consider that enlightening, and maybe that it was worthwhile to look into the black box. And in particular, I'd be interested in some personal perspective of him, or from him, as to the results that we have gotten, that I would tend to argue are in favor of a long-standing debate that was kicked off by Noam Chomsky's book criticism or book critique of Skinner's book Verbal Behavior in the 1950s. And so his perspective on these modern developments, I would also think would be extremely fascinating. My most favorite research activity is, of course, to do experiments and collect data. And then, if the data are digital, as they usually are, the next best thing is to write code so I can actually understand the data, to actually get some insights out of the experiments. And then, of course, the next thing is to present and discuss these data in our lab meetings and to hear from other people what they've done in the last week. So experiments, data, and all the things around that is the most favorite task that I can think of. And the least favorite, they have to do with reviews, and in particular grant reviews. So writing reviews for theses is quite tedious, because one has read the thesis and would know what's in it, and then one has to critique the thesis, which is sometimes, well, it's a tedious task. It has to be done, but it's a tedious task, in particular when one can't just write what a fantastic thesis it was, what a great student, what great work, and be done with it. And then all the burden around writing reviews for grants, and actually reviewing reviewing the grants as well. So reviewing grants is tedious and a chore, because I don't think I've ever met an experiment that wasn't worth doing. So I'm probably your most desirable grant proposal reviewer, because I like all the experiments that are being proposed. It's very rare that I think some experiment is not worth doing. And so usually very quickly one reads something, okay, we want to look at this and that on the first page, and you go, yeah, this is really interesting, one should really do that. And then the rest of it is really just, okay, how are they doing it, what methods are they using, and then try to find something that may be wrong with the method, when science is all about failing, and science is all about trying it out and see what happens. And so, of course, one can see this as an opportunity to help colleagues devise, maybe come up with a better strategy, but I mean, who am I? I have some experience, but very often the craziest experiments are the ones that nobody would expect yield any reasonable result, and then they come up with something totally amazing that one would have never thought of. So these grant reviews I always find very difficult to do, and it's usually always an exercise in finding something wrong that one really doesn't want to find something wrong with. And then, of course, once one has read the grants, and then one has to write all this and formulate that in a way that is constructive and where it doesn't show too much that I really had a very hard time writing the grant review. So those are the best and the worst things about this job here. receiving reviews and decisions is clearly by far the most stressful thing in our line of work. Sports, family, and hobbies. Pretty much always in that order. It's necessary and important, but not always necessarily fun. Yes, all the time, and we did so even before it was cool to do so. Yes, I remember the title of my first article. If every article counts, it's my first article published in a scientific journal, and it's a review article that we had to write as undergrads as a part of a course. And its title was Chaos, Cheating and Cooperation, Potential Outcomes of the Iterated Prisoner's Dilemma. And it was a review on the Iterated Prisoner's Dilemma, a theoretical model within the field of game theory to try to explain the evolution of cooperation. Well, while the first article was easy, the second is more difficult because there are more and more articles accumulating. But the latest article that we made public, as in putting on a preprint server, is the work called Identification of FoxP circuits involved in locomotion and landmark fixation. And that's work, the most recent experimental work on using CRISPR-Cas9 to do some genome editing in the fly to find out more about the function of FoxP, of the FoxP gene, and what it does in behavior, development, and learning of the fruit fly. Well, by the full definition of open access, not all my articles are open access, because it also means they have to be published with a liberal reuse license, which probably some of my articles are not. Some of them have been made open access at some point because they're already old. Some of them have preprint versions or other copies that are available. At the very least, all my articles are available from my website. So they're accessible, many or most of them even maybe open access, but probably not every single article I've ever published also has a liberal reuse license, which probably isn't such a big deal given that what I do is not really commercially relevant. I think I'm most proud of a research result that came after more than a dozen years of research, of my own research. And it, to my understanding, solved a question, a research question that was formulated, to my knowledge, the first time in 1934. So it took me with a new approach that people hadn't been trying before. It took me more than a decade to develop the right experiments to answer that question, and that is the distinctiveness of the genetic mechanisms underlying operant and classical conditioning, respectively. So what we now call world and self-learning. And so that these two processes have different biochemical and biological, neurobiological mechanisms underlying them, and how we're starting to understand how they interact. I think that's something that I'm very proud about. It also means, because that was now more than 10 years ago, this also means that I'm, for my own ambitions, way overdue with another thing that I should be proud of. So, or equally proud of. We make things every year, we find out things every year that I think are pretty cool, but there's cool and there's cool. So I'd really like to make a discovery again on that order of magnitude, and I wish it would be so easy to control that, and that one could make the discovery on demand rather than the discovery coming to you. Doesn't everyone want to go back in time and tell their younger self something about the opposite sex that they've learned in the meantime? No. But I'm extremely fortunate and privileged in a lot of ways in how things have turned out. And I think if I would change anything, I'd just screw that all up. So, no. I would change nearly everything. Let's just start with actually publishing something. If we had a publishing infrastructure from maybe 10 years ago or so, not even one from this year, just from 10 years ago, what we could be doing is we could just get together all the authors from around the world, write an article, put the figures in by dragging and dropping the data there, for instance, to get flexible data visualizations where the reader can play around with angles, can play around with certain data subset, whatever it is beyond of what we authors have selected. And then in order to publish, all the authors just have to click on publish, and as soon as all have clicked on publish, it will be public. And whatever happens then, one can talk about that as well, but that's what I would change in the publication process. And of course, that would be totally independent of publishers or journals or anything of that ancient sort that we're doing today that is totally antiquated. And nobody in their right mind would design a system we have today if something happened to the system that we currently have. In the traditional sense, as we're doing it since around the 1960s or 70s, I think peer review is important, but pretty much overrated. I would change pretty much everything, if I could, about peer review, if you mean the traditional way of peer review. For one, I would open it up to more than just a handful of selected individuals. Practically, I would let the reviewers comment directly on the document, and the document would look the way the reviewers design it. This means that we submit it in a way that is flexible enough so reviewers can review the way they want, on the screen, on paper, any way they want, to facilitate the peer review process just practically. I would make it interactive such that authors and reviewers could constantly interact with each other, because very often things happen simply due to misunderstandings, and very often as a reviewer I just have a question that can be easily answered. So in a lot of ways I would just bring peer review into the 21st century, because it's still stuck somewhere in the middle of the 20th. I had no idea. So I had to check my stats, and it says I have 110 total recorded reviews. So that's about 6 reviews a year, or half of a review by month, which is quite a bit more, about 4 to 6 times more articles that I review than that I publish. We definitely have to fight for open science. The reason is we can't wait for open science to just come around. Our institutions have had 30 years to modernize our infrastructure, and if one would have a modern infrastructure, the default would be open, such that you would have to go the extra mile and extra effort to make it not open. You still could make it open, but if you wanted to make it closed, you'd have to go the extra, put in some extra effort. Right now it's the opposite. If you want to be open, you have to put in some extra effort. And if open also means reproducible, because if something is open and we check it and we can quickly find out that it's not reproducible, it means that the science that stays around is reproducible, then there really isn't any way around open science. So since open science is just science done right, and our institutions are not providing us with infrastructure to do science right, then we have to fight for it, don't we? That question is a little bit like asking, is every picture that you take on your cell phone actually stored there? If we had a modern infrastructure for recording our data in our experiments, in our labs, then automatically every experiment that we do would be properly documented, because I couldn't do it any other way. There are some exceptions, maybe when you just sit down somewhere and you try something out, but that's, you just try to see, can the flies do this, how do the snails behave in this or that situation, when you are just checking something without actually really recording anything in any way, shape, or form, just to see if it's worth going further in that direction. But that's, I think, quite an exception. And usually our experiments that we do, they're designed beforehand, and we think long and hard about how to do the experiments. And then if they're all recorded automatically and the data is being made accessible, as they should be, given that it's the year 2020, then this would be automatic. We wouldn't even have to make a decision of whether to document and publish results or not. It would be automatic, and that's how it should be. Well, an experiment can ever only go wrong if the control groups don't do what they are supposed to be doing and what they have been doing over time. And so then what you have to do is you have to figure out what is going wrong such that the controls are not doing what they should be doing. Clearly, a lack of a difference between control groups and experimental groups is not an experiment gone wrong, that's a research result. And there could be any kind of reasons why a control experiment does not show the results the control experiment should be showing. It may be a measurement error, it may be a contamination, any kind of reason, then one just has to find those reasons and eliminate them. That sounds easy, but may be very tedious and difficult practically. Novelty without reliability is a little bit the consequence of publish or perish. Publish or perish means either you publish your articles, and today this means in many articles, as many as possible, in as high-ranking journals as possible, or get kicked out of science. Translated, it means, in today's practical terms, it means publish in journals that value novelty, irrespective of whether what novel things you claim to have found are actually science and actually reliable results. This is what most top journals are trying to do and are trying to incentivize by putting a lot of emphasis on novel and unprecedented, surprising results. And if one looks at the data of all the published research, not the retracted research that make the headlines, which are also, of course, all in the top-ranking journals, but if one really only looks at the published articles that are still published and that are not under dispute, then what one can find is that technically and methodologically, the higher up you climb into the journal hierarchy, the less reliable are the methods with which these results have been obtained. So novelty without reliability is in essence the most threatening practice that we currently have, that is most threatening to the scientific enterprise as a whole in terms of publicly funded scholarship. If the public is supposed to fund an enterprise such as science, then of course only if we can show that we're doing our best, our very best, to make sure that the science that we produce with these public funds is also reliable. Given that the current system in a lot of ways does the opposite, I would say that this practice is a threat to publicly funded science, as it undermines and actively undermines the public trust in science. If I were king or emperor for a day and I had the power to change just one thing, I would make sure journals cease to exist. Journals are the biggest obstacle to reform in science, and so I would love for them to just go away. In science generally, there isn't really anything that needs to be kept or maintained, because if you do an experiment or whatever it is you do and you find an improvement, whether it's a method or a finding, then you discard it. Newton was discarded when we discovered quantum mechanics and relativity. So I don't think anything really needs to be kept, perhaps other than at least a general form of the scientific method, But of course, I wouldn't exclude that at some point in the future we can improve also on the scientific method itself. And I don't think the scientific method has been all that constant through the last 3 or so centuries either. So if anything, probably science revolves around a general concept of a scientific method, but even that is probably up for improvement. I've received so much advice over the many years, it's impossible to pick out one that would be the best. Many of them, probably the very best and most helpful ones, I've internalized to an extent that I can't recall from whom I received it. But there's been one very memorable piece of advice that I received from David Glanzman from UCLA. And that was just so memorable because of the metaphor that he used to explain it. And he likened publishing to a bullfight, where the authors are the matador and the picadilleros, and where the bull is a metaphor for the reviewers and the editor. And the idea here is that the matador, and with the help of the picadillos in the neck of the bull, would try and do everything they can to prevent the bull from raising its head. If the bull can raise its head and see you, you're dead, you're gorged, you will not survive the bullfight. And so you have to write a paper to prevent the editors and reviewers from seeing the mistakes and the flaws that are in it. And that does not mean that you have to hide anything. You have to put everything there in plain sight, but write it in a way that everyone understands how you've done your experiment and sees that the flaws and mistakes that every work always has. It's a practical work. There's always there never is something that is absolutely perfect, but that editors and reviewers can see how and can put those negative aspects of the paper in contrast to the positive aspects. And this is both a true and correct metaphor and useful advice, but it also shows how, especially in today's day and age, word crafting and command of the English language, and in many cases storytelling, and in the extreme case questionable research practices when writing your paper, has become important, and where the lines between presenting your research and telling a story that isn't backed up by the data are starting to blur. And so, both because of the vividness of the metaphor, its truth in the current environment, and because of the questions that it raises with regard to what would happen if we all would go to extreme ends to prevent the bull from raising its head, I think that was a piece of advice that was very memorable and worth discussing, and is being discussed many times after hours. What keeps me going in science? I'm a news junkie. I want to find out new things. Anything that's new to me is interesting, and that's what keeps me going most of the time. I love what I'm doing, and that's all it is. It fires my dopamine neurons and makes me happy. Probably the best way to follow our research is the scientific literature. Then the second best would be on my blog and on Twitter, where we try to provide some background information. If you want to have a very tough way to follow our daily results, you can go to lab.brembs.net, where we publish our results, but most of the people in our lab are too lazy to actually explain the results that they have, because we use those figures in our lab meetings when we discuss what have we done last week and what are we going to do next week. And so in order to follow that, closely, one probably has to have a quite detailed idea of what's going on in our lab. Well, of course, my mentors, Martin Heisenberg, Jack Byrne, Randolph Menzel, I'd be very interested to see how they answer these questions. Oh, but then of course, if my wife is watching, then she should be, she should be answering those questions, obviously, first. Critique. The most important aspect of science is critique.